In the name of Allah, the most merciful and the most gracious. Students, we are doing chapter number 16, support and movement, and our today's topic is ultrastructure of the skeletal muscles. So here are some terms that I have mentioned here again, which you need to reinforce for the purpose or for the sake of the next topic's understanding. So sarcolemma is the specified or specialized plasma membrane which surrounds the muscle fiber. Sarcoplasmic reticulum is just like endoplasmic reticulum which runs into the muscle fiber. Myofibrils are the thread-like structures which are present within the muscle fibers. Now the actin and myosin filaments that we will discuss here in detail. Myosin are the thick filaments and actin are the thin filaments. Actin further contains two proteins called troponin and tropomyosin. Another topic here that we are going to reinforce is Z line, I band and A band, which we have discussed in the previous topic. I band is isotropic band and A band is the anisotropic band, which can polarize light and which cannot polarize light. And the Z lines, H zone, M line, these all things we have discussed in the previous topic. Now the sarcoplasm is just like cytoplasm of other cells, which contains the sarcoplasmic reticulum and which is basically the site for the storage of the calcium ions, and it also surrounds the myofibrils with the muscle fibers. Now here is the structure of a sarcomere in which M line is shown, the Z lines are shown, the actin and myosin filaments are shown. Sarcomere is basically a smallest structural unit of the muscle fiber which can cause movement, which can contract, which can become shortened and can cause the movement in the muscle fibers. Now if we talk about the thick filament and the thin filament, they are myosin and actin respectively, and their structures are very beautiful as they look like the beads which are coiled together. Thick filaments are myosin, they are 16 nanometer in diameter and they contain 6 polypeptide chains which are arranged in such a way that has a tail terminating in two globular heads. Now in the thick filaments of the diagram you can see the globular heads which are emerging out from the thick filaments, and they are showing up like the drops. Myosin tail consists of two long polypeptide chains which are coiled together, and each thick filament contains about 300 myosin molecules. These heads are responsible for the cross bridges which are formed between the thick and the thin filaments during the process of contraction. The transverse sections in these regions indicate that 6 actin filaments surround each myosin filament, and the movement will be done by the actin filaments because they are thin, and the myosin filament will stay in its place because it is thick. The arrangement of the actin and myosin results in a number of other bands being recognizable in the sarcomere. Now we will talk about the thin filament, that is actin, which are shown here with the blue bands or the beads which are coiled together. They are 7 to 8 nanometer in thickness, and they are composed chiefly of the actin protein. Actin proteins are arranged in two chains which twist around each other like a twisted double strand of the pearls. One is the globular actin or G-actin. It is kidney-shaped polypeptide subunit of the actin, and the fibrous actin or F-actin Now we will talk about the tropomyosin and the troponin, which are shown here in this diagram. The troponin are shown by the three red beads, and the tropomyosin are shown here in this diagram with the help of blue beads, or the strand which is blue in color. Tropomyosin are two strands of another protein which are twisting around the actin chains and helps stiffen its structure. In the relaxed muscle fiber, they block myosin binding sites on active, so that the myosin heads cannot bind to the actin filaments and the cross bridges may not form. Troponin is the other major protein in thin filament. It is actually three polypeptide complex. One binds to the actin, another binds to the tropomyosin, while the third binds to the calcium ions. Both troponin and tropomyosin help control the myosin-actin interactions involved in contraction. Now coming back to the sliding filament model. The sliding filament model has been divided up in 5 phases. First of all, the nervous impulse, it will arrive at the neuromuscular junction, which causes the release of a chemical called acetylcholine, which causes the depolarization of the motor end plate, which travels throughout the muscle. Now the calcium will be released from the sarcoplasmic reticulum, and it will surround each myofibril. The calcium which is released from the sarcoplasmic reticulum is basically responsible for the muscle contraction. Now in the muscle contraction, in the relaxed muscle fiber, the thick and thin filaments overlap only at the ends of the A band. But when the muscle fibers are stimulated by the nervous system, or when acetylcholine has been produced, the myosin heads are attached on to myosin binding sites on actin in the thin filaments, and the sliding begins. The A filaments will come closer. Now what will happen here? I band shortens, H zone disappears, Z lines are brought closer together, and A bands move closer together but do not change in length. Now how the cross bridges are controlled? Muscle contraction is initiated by nerve impulse that we have discussed. The nerve impulse is carried through the sarcolemma to the T tubule and then to the sarcoplasmic reticulum. And here the calcium gates of the sarcoplasmic reticulum will open up, releasing calcium into the cytosol. When muscle is at rest, The tropomyosin is disposed in such a way that it covers the sites on the actin chain where the heads of the myosin becomes attached. And when muscle is required to contract, calcium ions bind with the troponin molecules and cause them to move slightly. This attachment of the calcium ions with troponin has the effect of displacing the tropomyosin and exposing the binding sites for the myosin head. This whole process of muscle contraction or sliding of the filaments needs energy to take place, and this energy is provided by the ATP molecules. When ATP is broken down into ADP and the phosphate molecules. The formation and breakdown of cross bridges occur again and again during the sliding filament model. Here again the sarcomere shortens in the muscle and the muscle shortens ultimately. The release of the phosphate triggers what we call the power stroke of contraction, in which all the actin filaments will slide past the myosin filament just like the power stroke. This is because the ADP, which is still attached, causing the myosin heads to rotate, pulling the actin filaments past the myosin filaments and towards the M line. In phase 4, myosin then detaches from the actin, breaking the cross bridges as the ATP binds again to the myosin head. The whole process is repeated when the myosin head attaches to an actin binding site. Further down the actin filament, another power stroke occurs. One important point that we need to discuss here is the rigor mortis. That is the state when the muscles become stiff and no movement of the muscles can take place. And this is the state when the person is called dead and cannot show any movement of the muscle fibers because all the ATPs has been consumed and there are no more ATPs, which can cause the contraction of the muscle fibers and they become stiff. In phase 5, muscular contraction continues so long as there is ATP and enough calcium in the sarcoplasm. And if the ATP and calcium has been consumed and no ATP and calcium is left in the sarcoplasm, the condition is called rigor mortis. But if the nervous impulse stops, the calcium goes back to the sarcoplasmic reticulum and the actin returns to the resting position, that is, the muscle lengthens and relaxes. Now here are some questions related to the topic. You have to answer these questions. Thank you so much.